Hello, Science 30. This lesson is going to be on mutations and kind of a neat little picture that we have here, a comic that's showing these two birds that look pretty much the same, except, of course, what is different. If we take a look at the beak here, and if we take a look at the beak here, so the beak length, the beak shape is determined genetically. It's determined genetically by the DNA, the instructions in DNA that is used to make proteins, and those proteins in this case are responsible for the differences that we do see here. So within this population of these birds, kind of the key is that we do have what is called variation. So just like within our population, the human population, there is variation in many, many different ways. Some visible, like we see with these birds, other ones not visible. And the source of that variation is DNA and changes to the DNA, and that is what is referred to as a mutation, an alteration to the code, the A's, the T's, the G's, and the C's in the DNA, and in turn changing the proteins that are produced. So if we do take the DNA and alter one or more of the, so it is the nitrogen bases, remember, that do code for the genetic information. So DNA made up of the sugars, the phosphates, the nitrogen bases. The nitrogen bases, yes, they are the A, the T, the G, and the C, and that is the genetic code. So whatever is, well, what I have in quotation marks here, the normal version, if we do have alterations to the genetic code, then we do have gene variation, and that gene variation can alter the structure and the function of the proteins that are ultimately produced. So just as a reminder, it is inside of the nucleus that we're taking a look at here, where we do have the DNA, we have the genetic information. For Science 30, remember that you don't need to know about this one here, the RNA for Science 30, the messenger RNA, which is really the link between DNA and the making of proteins. As far as Science 30 goes, we go straight from the DNA and we use that code out in the cytoplasm to make, well, here it has it as a peptide chain, but that is really our protein that we are talking about. So proteins, yes, they're responsible for pretty much anything you can think of that is taking place inside of your body, inside of your cells. So whether it is digesting food, whether it's contracting your muscles, whether it's transporting oxygen around in your body, it is proteins that are carrying out all of these functions, not the DNA. The DNA provides the code for making the proteins, but it's the proteins that are really doing all of the work. Proteins are made up of amino acids, so if we do change the amino acid sequence, it's not 100%, but it may alter the ability of the protein to function properly. Sometimes just changing one single letter in the DNA is enough to change one critical amino acid, and that's enough to alter the function of the protein. So in fact, it may not work at all anymore, or it may not work as good as what it used to. So whenever there is a change again to the DNA, that is what is referred to as a mutation. So if we do have a protein that no longer functions anymore, that's probably a negative mutation, but there are other mutations that can actually be beneficial. So if we go back to that first picture, perhaps the birds within the population started off looking like this, but then there was some food that was available, but it required a longer beak. So it's not the requirement to get that food that allowed the beak to grow longer. This is a genetic trait. But due to mutations in the DNA, some birds may have been born with a longer beak, and that would give them a selective or beneficial advantage. A lot of the time, though, when we're talking about DNA that's coding for proteins, we might have the nitrogen bases that are hundreds or thousands of letters long. So a lot of the time changing it actually doesn't have any effect at all in terms of altering the structure and the function of the protein. But a couple of neat ones at the bottom here. So this is an actual picture of a hummingbird, and we can see that, yes, in some cases, so this beak here looks like it's probably about 10 to 15 centimeters long. So that is due to mutations to the DNA and natural selection, an advantage for this bird to access a certain resource, which is probably pollen in a flower in this case. So it allowed those individuals to survive and reproduce and pass that genetic trait on to its offspring as well. This fish that we're taking a look at here, kind of a unique fish. It is called an ice fish. They are found in the Antarctic waters, and kind of unique because vertebrates, they all have red blood cells, but these ones here, they actually have no red blood cells. So their blood is actually more or less clear in color, and that's kind of like the fish as well, almost transparent, because there are no red blood cells, there is no hemoglobin. So they're able to transport oxygen because of the very cold environment, and more oxygen is able to dissolve in cold fluids. So mutations, why do these mutations actually take place? Well, there are things that we are exposed to in the environment that do cause changes to the DNA. So exposure to ultraviolet radiation from the sun can cause mutations to our skin cells in particular. Fortunately, in our upper atmosphere, in the stratosphere, we have ozone, or O3, and that filters out most of the ultraviolet radiation that is coming from the sun. Environmental toxins, they can also cause mutations. So like we're seeing in this picture here, pyrobenzene in cigarette smoke, X-rays, radiation, the other picture that we do see here, a higher energy radiation than ultraviolet radiation, and that can cause mutations to the DNA. But a lot of the time, remember that every time a cell divides through mitosis or meiosis, you need to replicate the DNA. And this is a lot of bases to replicate. On the human, 46 chromosomes. Remember, we're talking about 3.2 billion of these bases or steps, and that's actually only on one set of the chromosome. So yeah, we even have to multiply this by 2, and we end up with a lot of these letters that we need to make exact copies of, but this just simply doesn't always happen. There are mistakes that are going to be made, and these mistakes are mutations, and they can lead to changes in the proteins that are produced. So occasionally, if the mutation does occur at a strategic point on the gene, it may alter, yes, the protein. So again, a lot of the time it has no effect. It really depends upon where that mutation is. So if it is in kind of a critical point that I'm just calling here a strategic point within the gene, then it may have very detrimental effects and even be deadly in terms of that individual if they're unable to survive without that protein, if it is that critical of a protein that is necessary for life to exist. Somatic cells are all of the cells in your body except for your sperm and your eggs. So sometimes we just refer to these as your body cells. And these, of course, are the ones that are diploid. And in humans, the number is 46 chromosomes that we're talking about. So if you are exposed to ultraviolet radiation, for example, from the sun, if it does cause mutations and damage to the DNA in your skin cells, those are your somatic cells, and that is not passed on to your children. However, if there are mutations to egg and sperm, and the zygote as well, which results after fertilization, this is the source of all of the cells for that individual from that point on through the process of mitosis. So remember, we started off with the one fertilized egg, or the zygote, and if there is already a mutation to DNA in the zygote, then when it divides into 2 cells, each of those will have those mutations. When it divides into 4, so all of these cells from that point on will now all have those mutations that arose in the original cell, whether it was the sperm or the eggs. Very important to realize, though, again, going back to that very first picture, the reason why we are all different. And variation is critical within a population. We want to have a great deal of variation. And in fact, in the human population, what they do say is we actually don't have a huge amount of variation. But it is mutations that are the ultimate source of all variation that we do have. So if you think about visible variation, hair color, eye color, all of those variations are due to mutations. In the case of hair color or eye color, of course, Those ones don't really provide us with any selective advantage, but there's other variation as well that we can't actually see. For example, some people might be able to digest the sugar lactose that you find in milk, and others may not. So that is a variation that we can't tell just by looking at someone, but that is biochemical variation. We're going to talk a little bit more about bacteria in a bit here, but under stress, various different kinds of stress from antibiotics is one example. Bacteria, their repair mechanisms to their DNA, it actually decreases, and their mutation rate bumps up considerably, maybe up to 10,000 times higher. So this doesn't result in mutations specific for the antibiotic that they are exposed to. However, because there are more mutations, it increases the likelihood that maybe at least some of those bacteria will develop a resistant gene, and that is what actually leads to then antibiotic resistance. So types of mutations, important for you to know these types of mutations. I have a very nice clip here that I'm not going to play for you, but just one segment of a PBS Nova series, if you want to take a look at this. PBS or Nova, and it's called Cracking the Genetic Code. Cracking the Genetic Code, and just this one segment, one wrong letter, what it does actually talk about is mutations to the DNA, and it talks specifically about an example of what is referred to as Tay-Sachs disease. And that one actually ends up being a fatal condition, Tay-Sachs, where people die at a very, very young age. So 3 different kinds of mutations that you should know about. So the language that we do use, the language in your textbook, this first one here, they are called either substitutions or a point mutation, because it's only one single point, one letter in the DNA. So this alters, yes, only one base, only one letter. Now keep in mind as well that there are 2 sides to the DNA double helix. So if it changes this A to a T, then it is going to have to change the base that is complementary to it as well. So it's really the entire step that is being changed when we do have a substitution. So if that is the case, there's potentially a maximum of only 1 amino acid within that protein that could be altered. So this is what it looks like. So here we do have a segment of DNA, and remember that we can sort of group these bases into 3 at a time, and those are what we call the triplets. And it's these triplets that you would use when you're reading your DNA triplet table. So just as a reminder again, in reality, there is this, well, a messenger RNA that is actually in the middle here. But for Science 30, we don't worry about that. We just kind of skip over that. So what we say is that the triplet in DNA ATA codes for the amino acid tyrosine, CGG for alanine, GAG for leucine, and CCC for glycine. Those are the 4 amino acids. And it's a 3-to-1 ratio, right? So for every 3 of the bases, we end up with 1 single amino acid. So what happens if we change the 3rd adenine to a thymine? I think I probably meant the 3rd triplet. The adenine in the 3rd triplet to a thymine is what it looks like here. No, sorry, it is the 3rd adenine anyway that we do have. This one here. So what happens if we take this one and we change it from an A to a T? So changing that triplet, if you now go to your codon table, and if you take a look at this one here, it's no longer leucine, now it is histidine. So we've changed 1 amino acid. So is this a big deal? Well, sometimes proteins can be hundreds of amino acids long. So changing 1 is maybe not going to make a difference, but it really does just depend. Is it in one of those, again, what I called strategic or critical locations? And if it is, what you might end up with is a dysfunctional or not as functional in terms of the protein. So that is substitutions, changing 1 letter, changing 1 step in the ladder, and changing 1 amino acid in the protein. So yes, depending upon where it is, that really depends upon whether or not you're going to get a protein that is going to end up functioning in the end. But most of the time, because it is one single 1 point for most of these, it's probably not going to have an effect. And also keep in mind as well, we talk about what's called the redundancy of the genetic code. Sometimes you can change one letter in the triplet and it actually codes for the same amino acid. So other kinds of mutations, deletions means you're removing one of the steps. Insertions means you're adding an extra one in. And in fact, I think in your textbook, they actually call them additions rather than insertions, but usually they're referred to as insertions. And what this does is it changes what we call the reading frame or the frame shift. It shifts all the other letters over either to the right or to the left. So again, for you guys, you don't need to worry about the messenger RNA. We would then say for Science 30 that it changes the reading frame of the DNA. So now, potentially all, not just that one amino acid where that base was removed or another one added in, but potentially all of the amino acids after that point could be affected. So our example here, same DNA sequence that we have, same set of 4 different triplets, 1, 2, 3, 4 DNA triplets that we have. These are our original amino acids. Now if we have the insertion of an adenine that we stick in here, so I stuck it in after the first triplet, and that's going to shift everything over. So the first of the triplets is still ATA. It still codes for tyrosine, but now all of the other ones have shifted over a little bit in this direction, and we end up with this extra one at the end that then wouldn't code for anything. So notice that now every single one of those amino acids from that point on are going to change. So these deletions or insertions, as in this picture here, these are frame shift mutations, and they have much, much more dramatic effects on the functioning of the protein than the previous example, which were the substitutions. So we'll take a look at a case study here. Already mentioned the antibiotic resistance. Huge problem in medicine. Other forms that are similar to this, herbicide resistance, pesticide resistance that we do have in crops, it arises in a similar fashion. So just as we have variability, so do bacteria, so do pests, insects, so do plants have variability as well. And this variation, once again, it results from mutations to the DNA. So variation in terms of bacterial's ability to resist antibiotics. How did that actually happen? Now, we had antibiotics that did work, the first ones that they started to develop in the late 1920s. How is it that somehow these bacteria develop this resistance to the antibiotics? So it's really important to realize that it already exists prior to the use of the antibiotics. The antibiotic resistance did not cause variation where some bacteria were resistant. That resistance, it was actually already present within the population. So what do antibiotics do? Well, they kill off the bacteria that don't have the resistance gene, but the ones that do have the resistance gene, they now have an advantage and they survive. So those bacteria with that resistance gene, not only do they survive, they reproduce, and now as the generations continue on, they transfer on that resistance gene to other bacteria, and eventually more and more of the bacteria within the population are now going to have that resistance trait. There's another method that bacteria can also pass this trait on, and we'll talk about this in a couple of slides, something that, again, you do need to know for Science 30, and it is called transformation, but we'll see that again in a couple of slides here. This slide here, though, very nicely taking a look at, and this is antibiotic resistance. So within this picture here, all of these blue ovals that we're taking a look at, these are bacteria. And as it says at the bottom, these are normal bacteria. They don't have a gene that allows them to resist the antibiotics. But already within that population, before antibiotics were ever used, antibiotics, there were already, well, here it shows one, but very few. It might be 1 in a billion, it might be less than that actually have a resistance gene. So prior to the use of antibiotics, it really wasn't doing this bacteria any good in any way whatsoever. It didn't have any sort of an advantage. But as soon as we start to use the antibiotics, now all of a sudden, that bacteria has an advantage. The other ones, if they're exposed to the bacteria, they end up dying. That's what antibiotics do. Some of them might just escape the antibiotic. They may not be exposed to it. But this one in the middle here, that's the one that has the resistance gene. In other words, the antibiotic doesn't work. So what that also means is this one here survives and it gets to reproduce. So in the next generation, what we see is that there are many, 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 many more, obviously, that are going to have that trait and fewer that are going to not have that resistance trait. So eventually what happens is pretty much the entire population of those bacteria are now resistant to the antibiotic, and now this antibiotic is of no use whatsoever anymore. And in fact, unfortunately, this is exactly what seems to be happening in medicine. Of the dozens of different antibiotics that we do have, many bacteria have developed resistance against some of them, and there are a couple of bacteria that are completely resistant to any of the antibiotics that we currently have. Bacteria can do something else as well, and yes, so you do need to know this process here specifically, bacterial transformation, but all of these ones, they're kind of similar. So bacteria do not have sex. They don't have sex because they don't produce any sperm and they don't produce any eggs. So they more or less divide by a process that's very similar to just mitosis. It's not exactly the same as mitosis, but they don't have sex. But one of the things that can happen is bacteria, even bacteria that are from different species, they can actually pick up, they can acquire DNA from the environment. So if this donor cell bacteria happens to die and DNA is released into the environment, this recipient cell can pick it up. And if that happens to have the antibiotic resistance gene, then this cell, this bacteria, now just acquired that gene by picking it up in the environment. So other ones, again, they're kind of similar. In the one in the middle, it's just the bacterial virus, which is called the phage, which is transmitting the virus that we can see here from one bacteria to another. Sometimes bacteria, they do get together in a process called conjugation, and they form a little connecting tube between the two, and then we can have this plasmid that contains the resistance gene that's traveling over from the donor to the recipient cell. But overall, just kind of treat all of these as a form of transformation whereby the bacteria can acquire a new trait, and it's not acquiring the new trait due to a mutation. It's not acquiring the new trait due to sex. It's acquiring the new trait simply from picking it up from other bacteria through various different mechanisms, but it is coming from other bacteria ultimately. So this once again can lead to variation within, in this case, the bacterial population. This sort of thing doesn't take place with plants or animals. It's strictly going to be bacteria that we're talking about.